Buongiorno a tutti, grazie agli organizzatori.
Good morning, everybody. First of all, I would like to thank the organisers for having invited me. I'm going to illustrate the preliminary findings of a neuroradiological study that we are involved in. I would like to start by clarifying the role of PCDH19. I will skip the clinical introduction because it's something that we've already looked at. We know that we're talking about encoding protocadherin 19, a calcium-dependent cell adhesion molecule involved in neuronal circuit formation during development and in maintenance of normal synaptic circuits in adulthood. The expression of non-clustered protocadherins is linked to the basolateral part of the amygdala, the interoral cortex. In recent years, a number of studies have been carried out to clarify the role of PCDH19 in brain development. We have seen, for instance, on zebrafish, studies that demonstrate that PCDH19 is involved in migration and formation of neural circuits, and in mouse models we've seen that it is involved in the progeneration of cells. We know that this is also a fundamental protein in determining adhesion of neurons. We know that PDH19 proteins interact with the same cells that have the same types of protocadherin on the surface of the cell. In the case of mosaic disorders correlated to this condition, there are two different populations. This leads to an alteration of adhesion affinity, and therefore there is an intracellular adhesion alteration. We know that human-induced pluripotent stem cells are used to characterize PCDH19, and we see that there is a control of polarity during neurogenesis. This is important for correct neurocortical development. We've also observed that PCDH19 is correlated, cells with mutated PCDH19 have an advanced degree of maturity, and this leads to cell populations at different stages of neurogenesis, and consequently this leads to an alteration in brain development. These are studies on experimental and mouse models which have shown that this molecule is fundamental in the development of the brain, but MRIs of patients with this mutation appear to be within the norm, as we saw in the previous presentation. A number of rare cases have been described of malformations in these patients. We have a paper here which describes 5 patients, one with periventricular nodular heterotopia and 4 with FCD in the temporal central frontal regions. This other paper describes 4 patients with cortical defects and malformations. The aim of our study is as follows. Firstly, to investigate the presence of an abnormal brain structure, either morphological defects that are visible using standard MRI, or minimal defects which have to do with the gyration or thickness of the cortical region. And then the other aim is to evaluate the clinical outcome of a cohort of patients with PCDH19 gene pathogenic mutation. We have 20 patients, 18 females, 2 males with PCDH19 pathogenic mutation, multicentric selection, Meyer, Hospital, Bambin Gesù, Gaslini, and Stella Maris. We have acquired clinical data retrospectively with an ad hoc form that was filled in by the attending neurologist, with seizures onset, frequency of seizures at onset, follow-up, psychological, neuropsychiatric, genetic information was also collected. With respect to the focus of our study, i.e. neuroimaging, we carried out an MRI on 11 of our patients. We then decided to broaden our study to include another 9 patients involving our colleagues from the Bambino Gesù Hospital of Rome. We kept the breakdown of patients in order to avoid errors due to possible non-uniformity of data, since the patients were examined with 2 different machines. Ours is a standard protocol with 3DT1 weighted brain gradient echo for morphometric analysis of the cortical thickness and gyration index. We also acquired for 2 patients two sex agenda-matched, age-matched controls. These are the clinical results. The mean age at the time of study, 17, with a very broad range from 3.7 to 44 years of age. With respect to epilepsy, onset 10.8 months, with a range up to 2 years and beyond in terms of onset. Focal seizures during fever, 90%. 80% in cluster. We also have observed other follow-up tonic seizures, GTCs, absences, and focal seizures. 40% suffered from status epilepticus. We observed the EEG carried out during the last follow-up and observed that 40% of patients had focal abnormalities, predominantly in the frontocentral temporal regions. From the point of view of treatment, we did not carry out a specific statistically significant effective analysis, but observed that most of the patients were treated with valproate with benzodiazepine in association, which was considered as effective by clinicians. From a neuropsychological point of view, the development was normal in all patients before onset of seizures. In the follow-up, we observed that 70% of patients have intellectual disability from mild to moderate. The neuropsychiatric profile was characterized as follows: 55% autistic features, 63%. We have autism spectrum disorder diagnosis, whereas in 37% we have the presence of autistic traits. We found that in 35% of patients, we have other disorders, hyperactivity, attention deficit, obsessive traits, versus the 55% with autistic features. 10% were normal mutations, 70% missense, 10% frame shift, inheritance, 80% de novo. 2 twins inherit this from the unaffected father. 2 other patients from the mother. 1 is affected, 1 is unaffected, with borderline cognitive level and a case history of epilepsy. We decided to stratify patients by age because symptoms evolve over time and because the age of our patients was very different. The yellow curve indicates the presence of epileptic seizures. We saw the onset 0 to 6, 6 to 13, 13 to 20, and more than beyond the age of 20. If the onset the onset moment between 0 to 5.9 years, we see that we have about 100%, and then this figure drops when the onset is beyond the years of 20. We see that we have intellectual disorders patients over the age of 20, and we have these neurological disorders present in all patients for whom onset occurred after the age of 20. These are the particular aspects of the cohort. 2 pairs of monozygotic twins with an identical phenotype, and there's another patient with a monozygotic twin not included in the study because of the non-availability to carry out MRI with a discordant phenotype. The patient who was not part of the study is in a phase of remission, does not have neuropsychiatric disorders, cognitive levels within the normal. The one included in the study still has seizures every 3 months with mild intellectual disability. 2 males with mosaic mutation were included in our study. MRI imaging study details 2 groups of patients. I'll show you the data for the first group. The second group data is still being processed. The first group. the 11 patients who underwent MRI at Meyer Hospital, we used a neuroradiologist to examine the MRI. 7 had normal MRI, 1 had dysmorphic hippocampus with a temporal calcified nodule, 1 retrotrigonal and periventricular white matter abnormalities, 1 peritrigonal white matter abnormality, 1 cyst of the velum interposition. The second group had patients with normal MRI. We processed data to calculate cortical thickness and the gyration index. I'll show you an example of a morphometric study on a single patient to clarify our approach. This is the image with respect to the 2 healthy controls who were age-matched and gender-matched. The top part shows the left and right hemisphere, the medial and lateral sections in red and in yellow. We see an increase in cortical thickness. In this case, this is a patient with a more severe degree of alteration, the most severe included in our study, quite widespread, especially in the frontal lobe and the anterior region, but we know that these anomalies are present throughout the brain. The bottom part of the first chart indicates the calculation of the gyration index, and then we have the age-matched controls. In this case, frontal temporal lobe, and also the posterior part of the brain. What really matters is the group analysis. If we compare the entire group of patients with PCDH mutation with the healthy controls, these are the results. This is cortical thickness data. As you can see, there is quite a widespread thickening of the cortical regions in the temporal lobe, frontal pole. So there is no specific pattern with respect to the variations in cortical thickness in these patients, even though there's an alteration. The most interesting finding has to do with the gyration index, because as you can see, there is a very specific alteration which has to do with the temporal lobe, the parahippocampal convolution, and the gyrus. These patients versus healthy controls have a simplification of gyration in the areas that I have just indicated. Finally, we can conclude that we can confirm the known phenotype of patients with PCDH19-related disorder with early onset and seizures to decrease over time, especially after puberty. And often we observed remission beyond after the age of 20, but intellectual disability and neuropsychiatric disorders remain, especially autism spectrum disorders. With respect to neurological studies, our suggestion is that PCDH19 mutations can be associated with submicroscopal anomalies in the frontotemporal regions, hippocampal regions, and cingulate gyrus, confirming the role of PCDH19 in brain development in humans. I would like to end by thanking Professor Gorrina, Professor Marina, Dr. Lange, and all of the colleagues from in Gesù Rome, Dr. Speck, Dr. Trevisano, Dr. Longo, Dr. Napolitano, and thank you for your attention.